Hi, everyone. Today, we're thrilled to have a very special guest with us, Dr. Goldie Oza. He is a professor in Centro de Investigación y Desarrollo de Tecnológico de Química en Electroquímica, DITEC, and has an FMI too. He currently is working in cancer nanomedicine. His area of interest is the identification of cancer cells using magnetic resonance imaging, giant magnetoresistance, fluorescence, surface enhanced Raman spectrometry. In addition, he's also involved in tracking cancer cells by MRI and then specifically killing them by using antibodies, directed drug delivery, and including organ on a chip and nanomedicine lab at CDTech, which is a frontier science research. Lab on a chip is one area where he's exploring different types of magnetic and silica nanoparticles for the development of a sensor on a chip for prostate cancer detection and breast cancer detection. He has also received the UC Mexus Award for 2020 for electrochemical-based CO2 reduction in nanoparticles, complexes, and enzyme mining. He has published more than 80 articles and has more than 2,000 citations. He has written a book at CRC for its title, Nanotechnology Concepts and Applications, which was published in 2010. And he has edited a book on nanochemistry published this year in 2023. He has also won the first prize in the poster at Tech Nano 2018 for the detection of cancer cells in microfluidic systems. He has also given invited talks at different national and international congresses on cancer nanomedicine and cancer on a chip, thus promoting universal access to knowledge. He has collaborations at UNAM, Biomédicas, Tec de Monterrey, UNAM, Materials, UAM, Iztapalapa, Sinvestab, CIEL, Tec de Chetumal, University of Massachusetts, MIT Harvard Medical School, Italian Institute of Technology, University of California. Today, Dr. Goldie Oza will be sharing nanotechnology applications to human diseases. With his extensive experience and expertise, he will provide us with valuable insights and practical strategies that we can apply in our life and work. Please join me in giving him a warm welcome. Dr. Goldie Oza.
I'm very thankful to Professor Goldie. Thanks a lot for your kind introduction. And I hope this was too long, more than my presentation. Very sorry for that. So I think we can start with this session. I should follow that clock, right?
Yeah, fine. You're fine. There's no talk after this, so you.
Oh, that's fine. That's something great. So here we have. I would like to give you an introduction about the field of research which we are performing at the National Laboratory for Micro and Nanofluidics. So here the topic in which I'm interested in dealing with is the cancer on a chip platform for cell migration, immunotherapy, and nanomedicine. Now in this particular field, there's a greatest ever scientific warning which is being given to by Richard Feynman. He says the first principle is that you must not fool yourself, and you are the easiest person to fool. So that's the biggest scientific warning, and I hope everybody knows who is this guy, right? He's the Nobel Prize winner for quantum electrodynamics. So this, and of course, he's the, what do you call, I should not say the far, I guess, he's basically the biggest nanotechnologist, using for the first time in the University of Caltech in the year 1952. So then, now this person, why I'm talking about is based on his talk, we were interested in talking about nanotechnology and nanomedicine, because this was the talk wherein people were, I'm sorry, in the year 1959, that was talk given. So when this talk was given to people, they were not at all imagining about how these nanosystems can work. Now we have nanorobotics, but this kind of nanorobotic system was just virtually, I should say, it was just mentioned verbally by this guy. So you can imagine what kind of imagination he was having during that year when nothing was developed. He was talking about that the whole library should be on the tip of the needle. And yes, now the University of California has got that whole library on the tip of the needle. So that's something which he had imagined, and now it is coming up. So I think that's something which everybody must understand and imagine for now. And you being a student, I think imagination is something it's a powerful tool for a scientist. You think now, and it's going to happen in the next 50 years. So just imagine. Next, please. So we are talking about cancer on a chip here. So this was also an imagination which was given by many scientists coming from MIT, from Harvard Medical School, and there's a great guy. His name is Whiteside, George Whiteside from MIT. He was a person who started with this technology for the first time. And he said that, yes, why not? Why can't we develop a system in a three-dimensional manner? Because we know to grow a cancerous cell in a 2D system, but we were not aware of how to grow them in a three-dimensional system. Because our body is exactly in a three-dimensional form, and we would like to just not see the cells, the cancerous cells, the cancerous tissues in a 2D system rather than just talk about in 3D system, which is normally existing in our body. So what happened was people developed drugs, and those drugs were studied on animals, and people developed vaccines. Like, for example, the vaccine of we have the most dreadful pandemic which has happened, okay, for the 2 years, that is the COVID vaccines. So that was all developed, and it was used in animals, and thereafter that, who were the models? The humans were the model for them. Okay, so they didn't study in any of the 3D microfluidic system directly from animals. It just jumped on to human beings, and that's something dangerous, because that what happened later, most of the vaccines were not successful. Some of them were successful, even if they are successful, but there was so many side effects in there. And that was one of the reasons why we need the development of cancer or organ on a chip system, which is a three-dimensional model wherein the cells will be, in a space which is exactly mimicking the tissues of the body, the human body. And that's the idea. So here we have, let's talk about the cancer, but why we are talking about cancer? Because cancer, I know, I think everybody is aware of. The cancer is something which is creating a havoc. Women, the highest number of breast cancers, and men, the highest number of lung cancers. And they're creating havoc, and you can imagine in the next years to come, now at this particular age, it's like 19.3 million new cases have come up. But in the next, please, the next one, if you can see how it's growing at an enormous exponential phase. And in the year 2020, it's like 90.3 million. And then in 2040, it's just an estimate. It's just an estimate, but this can be, of course, more than 30 million people. And that's something which is very dangerous. We are moving in a stage. Why? Because we have so many food habits. We have so many incidences. We cannot avoid them. We have so many radiations coming from 5G, then comes 6G, then comes so many Gs. And then comes Goldie. So I'm very sorry for that, but then this study, because I just made a joke because there were a few people who are about to sleep. That's the reason. I'm very sorry for that. Okay, so now guys, so there are like 2040. You can imagine that the cases of cancer will increase exponentially. And this should not happen. Even if we would like to stop it, then this particular cancer, this thing should become like, a normal, it will become a normal disease in future, for sure. But then we need to have a medicine for that. And that medicine will be developed only when we have a proper model developed. And that should be a personalized model. For example, there is Goldie Oza standing here. I should have a personalized model besides me. Okay, this model is of Goldie Oza. This model means I'm talking about the cancer on a chip, organ on a chip model, like the gut system, any of the kind of chips which we would like to develop, it must be very much personalized to the person or the patient. So that is the kind of development we need at this stage. Please, the next one. So here we are talking about, the tumor and events in metastatic cascade. Now, what are the different kinds of metastatic cascades, and what are the existences of the cells? What kind of cells are existing? How there is this kind of stress which is being developed, because of which the metastatic cascade events they initiate or they start. Now, what are the steps? The first step is shedding. So what happens is the cancerous cells, they will move, they will grow, all right? They have a region wherein there is hypoxia, there will be acidic pH. So all these are the reasons which will make a tumor, okay, flourish, okay? And then after that, once a flourishing happens, then these cancerous cells will do the shedding, then there will be invasion, then there will be intravasation, and then it moves into the circulation in the stromal network. It moves into the circulation. Through circulation, it moves to eat in any organ, in whatever place they want, they will move ahead, and then finally they will extravasate out, and then they enter into different tissues, different organs. So this is what is metastatic event, it's a cascade of events going on. And this leads to primary, secondary, tertiary, these many different kinds of tumors in the body. Now the next one. So here we have, you can see here the same process, but in a very, very globular way. So here we have the invasion, the primary tumor, the invasion, intravasation, then they move in the blood, and then they will be moving then they will come out of the blood, gets into the tissue, and that leads to the metastatic site. So this is a regular process which is happening, which is continuously happening in the cancer patient. And people are not still aware why is this happening. What are the different kinds of intravasation or the invasion procedures by a cancer cell and how they spread? Of course, that's something which is not aware still, which people are oblivious of. So we need to solve this issue. And how is that? Of course, there are stem cells, stem cancer cells, and these are the origins. People have so many, thinkings or imaginations. Of course, we need to be very, very perfect in terms of those imaginations and then come out with a proper answer. That's the reason we need to have cancer on a chip system, wherein we can understand in the tissue which is grown outside in a more controlled fashion, that is it the stem cells? Is it something else? Is it that there is a tumor microenvironment which is creating this kind of metastasis? So what are the reasons behind this? And that's the reason that we have come up with this cancer on a chip system. Next one, please. Now here, we are talking about cancer on a chip model. Yes, we'll discuss about it. But then, before that, we need to talk about how these cancer on a chip or I mean, say the cancer cells will be detected, number one. Number two, when the drugs are interacting with these nanosystems, how these drugs with the nanoparticles will reach to a cancerous region or a cancerous cell. How we will target them. I mean to say how we will target the nanoparticles with the drug and get into the region of our target. Now, what is the, please don't get confused here, because my idea is to tell you guys that, and my aim of this research is first, we need to talk about the background about cancer. Number one, that's my background. Then the next thing is, what are the different strategies by which we can allow the anti-cancer therapeutics to get into the cancerous region? That is second. The third aspect is, once the nanoparticles with the drugs have been designed, have been developed, then we need to study in a normal cell line, I mean to say a 2D cell line, a 2D system, right? Then I wanted to show you that, okay, in the 2D system, how it works, all right? And then after that, I would like to show you that what happens in a 3D system, a 3D model. Okay, that's my, then that is the age, that is the point which I would like to explain to you guys. And then after that, explain you about how we can create or how we can mimic a complete organ in a 3D microfluidic system. And then study these nanosystems in the 3D microfluidic system so that we can now understand that how this nanoparticles with the drug are interacting with the 3D microfluidic system, and then how they are killing or how they are doing the job. Okay, and also we can track them inside the 3D microfluidic system. And once that is done, we can study also in human beings. So first study in the chip and then later do it on the human being. That's the idea. Okay, going back, I would like to explain you why I'm doing this shit by doing stuff. The thing is, what happens is whenever we go to a doctor, what happens? The doctor is giving you some prescription. I mean, let's talk about a patient, a cancer patient. He goes to a medical doctor. He has got a, he will have all his kind of reports, MRI and all those things, PET scans. And now the doctor is saying that, okay, my friend, you need this medicine, for example, doctors. But after that, what happens is, in next 6 months, of course, he will be given treatment every month. And after 6 or 7 months, the doctor feels like, oh my God, this doesn't work. This doxorubicin doesn't work. It seems his cells are not acting on this doxorubicin. I mean to say it's not working for him. Then after 6 months, then the doctor again changes his medicine, says, now I think I should give this medicine. So what is he doing? So he is trying to, he has the model, that patient as his own model. And he's just giving him medicines so that the patient who is there, okay, he has to, whether he is accepting it or not, he doesn't know. All right. And then after that, what happens? He's just studying, he's just making him as a model. And after the third time, I mean to say the patient dies. There is a very, very important discovery now, pembrolizumab. That's an antibody. It's immunotherapy therapeutics against a cancerous cell. It's a very recent drug, immunotherapy drug, which has come up. Now, people are so happy about that, okay, that drug is a wonder drug. It will solve all the problems of the cancer. That's something which people have told that all the types of colorectal cancers, all types of breast cancers, all types of, I mean to say the oral cancers, will be resolved by this pembrolizumab. But then, in the actual sense, when these drugs were given to the patient, what happened? There was necrosis. All right? With the first drug, there was necrosis with the cancer patient. With the second drug in the next month, there was complete, opening up of the vessels. And in the third month, the patient never survived. So pembrolizumab was very good drug, but only at the trials, in the clinical trials. It's a wonder drug only at the stage of clinical trials, but it's not a wonder drug. It's actually a suicidal bomb when it's actually given to the patient. So try to understand, this is what we are doing. This is a mistake which we are doing. We are blindly following the scientific journals. Oh my God, it has got this report, clinical trial, 100%. But how many patients are being used in that 100%? It's just 14 or 15 patients in that 100%. So we need to imagine, first we need to understand what is the actual meaning of 100%. Okay, we need to know how many populations they have taken care of. For Asians, it doesn't work. For Americans, it might work. For Latin Americans, it will never work. So this is the kind of, we have to understand the genetics behind, and all of you have studied well your population genetics, all kinds of genetics, I think everybody's aware of. So there cannot be a wonder drug for all the diseases. It's impossible. And especially in cancer, it cannot happen. So that's the reason we need to go for a personalized approach. A precision-based medicine is required. And that's the reason we need a cancer on a chip model. And here, so these are the zero-dimensional nanoparticles. Can we move ahead, please? All right, these are the different kinds of quantum dots. We are using these quantum dots, which are fluorescent in nature, and attaching the drugs onto the surface and tracking, in the body where these particles are going, and based on the fluorescence, we can identify them. Then the next one. Next. Thank you. Next. So here we have the gold nanoparticles, silver nanoparticles, iron oxide, copper nanotubes. So these are the nanoparticles which we have developed in our lab. We have made all of them in our lab to study. The next one, please. Now here we have a core shell system wherein gold is all on the surface, and this is iron oxide, which is like, this is a core shell body. It's inside is iron oxide, and on the surface we have gold. Now, what is the role of this? Why we have developed this kind of potent structures? Because we wanted to attach doxorubicin onto the surface. Doxorubicin is an anti-cancer drug. Now, this drug individually will be killing the cancer cell for sure. But we wanted to make this kind of drug to be more targeted. So we used some antibodies and then folic acids onto the surface, because we knew that the cancer cells overexpress folic acid receptors. All right, they overexpress certain factors, because of which we like CD44. These were the molecules which were overexpressed on the surface of cancer cells, and that's the reason we used antibodies against CD44, or we started using folic acid onto the surface, and also doxorubicin along with that. So this whole complex was then injected in mice. All right, in mice, number one, first of all, and later it was also inserted in 2D system, like two-dimensional cancer cells. So once these cancer cells or mice, in both of them we injected, if you can move ahead, please. Yeah, so these are the nanoparticles. These are the nanoparticles. Here you can see this is a cancerous cell. And here, how they enter, you can see these nanoparticles are located in these areas. They are, and the process, I think everybody is quite aware of the molecular biology. Like there is a endocytosis. There is a mechanism of endocytosis when the nanoparticles are taken up, endocytosed first, and then they enter inside the endosome. And then after that, in that particular pH, the endosome has got a pH of 4.5 to 5. Then from endosome, it enters into the lysosome, wherein the pH decreases further between 4 to 4.5. And then exactly at that point, the drug and the nanoparticles are separated because of the high pH, lower pH, they will be broken apart and the doxorubicin will be released. And now this doxorubicin, once it releases, it now then gets inside in the nucleus. And then they will attack the DNA topoisomerases. And then they will induce apoptosis in the cancer cell in general. So that's the mechanism of doxorubicin. Very simple, not to worry about it. But then next, please. These are these are the another different kinds of coherent nanoparticles. Can we go behind or back? Yes, here. This is a type of coherent nanoparticle wherein the core is made up of, this is the core, which is made up of iron oxide, and the shell is made up of gold. The next one, they have tried different magnetic systems. So here we have the core of cobalt ferrite and the shell of gold. Then next, we have another one which is manganese oxide, and then the shell, which is like a flower-shaped structure, which is of gold. Again, the next one. Yeah, please, again. Here you will find that this is a normal cell line, L6 cells, skeletal muscle cell line. It's a 2D system. It's not a 3D system at all. We have not entered into the era of 3D till now. We are just talking about 2D system. Now, what's the problem with 2D system is? 2D system has got a great surface area. If you talk about great surface area, means if the surface is like great, all the nanoparticles will be absorbed by them. All right. So it's a very effective system, efficient system. The 2D systems are very effective, and then they will attack all the nanoparticles will enter, the drugs will enter, and all of these drugs with the nanoparticles will be attacking from all the sides, and the cancerous cells have to die. You can see here, these are the cancerous cells, and there is like too much amount of apoptosis being generated. Okay, we have a targeted drug delivery here. Cancer cells against the cancer, we have the doxorubicin, which is anti-cancer therapeutic drug, and that drug with the nanoparticles, which are targeted against the cancerous cell, and they will induce apoptosis. Next, please. Then we also did hypothermia. So this hypothermia was what kind of hypothermia we're doing. Now, what happened is we wanted to check, all right, the drugs are killing the cancer cells, true. But then the second thing is drugs with the nanoparticles, they are not reaching towards the target effectively. And not only, even if they reach effectively, then what happens? These nanoparticles, if there is a solid tumor, which is very hard to be broken apart, if there is a solid tumor, these nanoparticles will not be able to enter. So what we did was we started using radio frequencies like hypothermia, heat the place, heat the region wherein there will be a temperature approximately of 42 to 43 degrees Celsius. And that particular temperature, it was easier for the nanoparticles to enter inside the cells, into the tissue first and then inside the cells. It was easier for them. So there was a two-pronged mechanism. One, we kill the cancer cell using this kind of drug, doxorubicin. And second, we're using heat to kill the cancer cells. So this was a two-pronged mechanism. Of course, I'm just telling you, yes, it's giving great result. This guy was like killing 90% of the cell. I'm just telling you that, in 2D system, it's just killing 80 to 90% cells are getting killed. Such a beautiful result you are getting. And then this complex, whenever, the next piece, this is flow cytometry, which was confirming that this is apoptosis generation. Apoptosis, I hope everybody understands. Yes?
Yes.
So apoptosis is programmed cell death. So this is a kind of mechanism by which the killing happens. Okay. So we confirmed using flow cytometry. All right, the early apoptosis, late apoptosis, and these are the numbers. Fine, fantastic. Next one. Then we injected in the mice. Okay, in the mice, we found the results are not quite appealing. All right, the nanoparticles, it's like this is a normal mice, and we injected the nanoparticles in the normal mice, and we wanted to see whether they're killing it or anything happens to them, how's the distribution of the nanoparticles in the body. So we did all sorts of measurements using magnetic resonance imaging. So using this magnetic resonance imaging, we can track the nanoparticles in the body. That's the idea. Next, please. Now, this is a tumor-induced mice. We had induced tumor, and you can find that this is a place wherein, where the tumor was placed, tumor was induced, and the nanoparticles you will find when it was injected initially during 0 hour, I'm sorry, here. So the 0 hour, there was not none of them. After that, in 24 hours, the accumulation happens, and later also in 48 hours, like almost it's like major number of things are there. So it proved that, okay, cancer cells have got some affinity towards the magnetic nanoparticles. All right, accepted. They have taken it, but they were not killed. The tumor size decreases initially. All right, next one, please. Then we did hypothermia also. We injected, now imagine, try to understand. We injected nanoparticles. We injected nanoparticles with a drug. And then also, so drug is doing the job of apoptosis induction, number one. And this is like injecting the needle or the probe, which is responsible for generating hypothermia. So what happened? All right. So here we have the mice, very bad health of mice, and we are very sorry for that mice. But then this guy he did all sorts of things, and I'm damn sure this got killed. I'm damn sure it was not killed because of the tumor. I think it should have been killed because of the treatment which we have given it. All right, we have given so much of torture to this guy. One, we injected the nanoparticles. The nanoparticles must have done a great job. Right, they must have reached those locations. Second, the heat which is being generated, that must also have created a good amount of pressure in the mice. And then finally, the mice died. Rest in peace. Next. So here, intelligent tumor microenvironment responsive nanoparticles. Now here, of course, all those previous research was also mine. I'm not going against my own research. I'm sorry. I would not do that. But the thing is, I myself, asking questions to me to like what's happening? Why is it in 2D, in 2D system, whenever I'm working on cancer cell lines, I'm getting a great result. But whenever I'm studying the same thing in a mice, simple mice, and we have induced tumor in that, we have done injections of tumors, for example, for almost like 3 months. And then after that, we got a tumor size of approximately 2 mm. Okay. And then we injected the nanoparticles into it. And there was the size decreased initially, like from 2 mm, it came down to 1.5 mm. But it doesn't work. It doesn't work 100% if it was not that effective. So why it's not working? That was my question. That means the results which I have got, that's published article. It's published in Nature, in scientific reports. So that's a published article. And I'm going against that because, come on, I'm not getting an answer for that. And I need an answer. And this answer is required because we are not getting answers for treatment of cancer. That's the reason we need to ask this question. So I have published, but I don't have answers. So that's the negative aspect. But then we are in search of this. Yes, thanks a lot for this. So now the next thing is we wanted to see that whether these magnetic nanoparticles are activating immune cells or not. Because, all right, these magnetic nanoparticles are killing the cancer cells, true. But then whether they are activating immune cells or not. Because if they activate immune cells, now I think everybody is aware of a little bit of immunology. Little, I'll explain. Don't worry, I know that the third or fourth stage, but I'll explain you a bit. We are all aware of T cells and B cells, okay? Because of COVID. Pandemic, as explained, is too much of immunology to us. So anyways, this T cells, there are certain kinds of T cells, and T cells means activation, right? Now, there are two types of immunity existing. One is, anybody? One is innate immunity, and the other is adaptive, right? Not adoptive, adaptive, right? So one is innate and the other is adaptive immune response. So in the innate immunity, neutrophils, macrophages, all of them present, of course, neutrophils, macrophages, basophils, eosinophils, all these come in the innate immune response. Dendritic cells, all of them. And the adaptive ones are T cells and the B cells. All right. T cells are responsible for making cytotoxic T lymphocytes, as well as they are also responsible to make T helper cells. And the other one is B cells. B cells are responsible to make antibodies. Okay. So now, what happens is if there is a cancer, so then if there is a cancer cell growing, then our immune system, the innate immune system, will detect them. And what they will do, the innate immune system means dendritic cells as well as macrophages, right? They will detect them. And once they detect those cancerous cells, after that, what they will do, these macrophages will give information to T helper cells. All right. And then those T helper cells will give information to the B cells. Then in T helper also, there are two kinds of T helper cells. One is T helper, which is responsible to activate B cell, and the other is T helper, which is responsible to activate cytotoxic T lymphocyte. Okay, not to confuse much, finally, just to tell you, there is a cancer cell activating macrophages, and macrophages activating a T helper kind of cell. T helper will activate cytotoxic T lymphocyte. And then this cytotoxic T lymphocyte will kill the cancer cell. So this is a cycle. All right, I will come again. So there is a cancer cell, macrophages detects it, T helper cell helps it, helps it to detect, and then this T helper cell will activate the cytotoxic T lymphocyte, and those cytotoxic T lymphocyte will come back and kill the cancer cells. So that's the cycle. So now what happens is you know this macrophage, right? I told you this macrophage. Now this macrophage, this guy plays double heel. There are two kinds of macrophages. One is M1 macrophage, and the other is M2 macrophage. Now, M1 macrophage is a very good guy. He's a good macrophage. He helps to kill the cancer cell. But M2, he is a double-edged sword guy because he helps the cancer cells to grow. He helps to invade. He helps to metastasize. So this macrophage M2 is the culprit. We need to punish him. And how will we punish him? By using nanoparticles. Then I thought, oh my God, here's the role of nanoparticle comes into picture. Next, please. What happened was, the next thing what we did was, there was an article in Nature Nanotechnology in the year 2014. And we could find that magnetic nanoparticles have a habit of activating, especially M1 macrophages. Because before that, I was not aware of that what is the role of magnetic nanoparticles exactly. So then with that article by, there was an eye-opener thing. And then I understood that M1 macrophages will get activated when the nanoparticles, that to magnetic nanoparticles help are existing in the body. So we injected these magnetic nanoparticles in the medium again, in a 2D system again. If you can go back, please, need to go back one slide here. So here, this is a 2D system again. I'm very sorry, we have to take the help of 2D every time. Okay, so here, this is it's a transwell system. It's a transwell plate. So in this plate, we have two compartments. At the bottom, we have the cancerous cells, and then this is a porous region, and on the top we have this macrophages. We started growing, especially raw macrophages. They're known as raw macrophages, cell lines of that macrophage. And then when this is growing, we injected nanoparticles with the drug and induced apoptosis in the cancerous cell. As soon as apoptosis was induced in the cancerous cell, then these guys, they got activated. They started secreting some cytokines like tumor necrosis factor alpha, interferon gamma. I mean to say these were the cytokines which were secreted. And moreover, these nanoparticles, the cancerous cells who were about to die, they were also secreting interferon gamma. And what happened? These macrophages, they started, I think there's a process wherein they do phagocytosis kind of process. So in that process, what happens? They have the habit of squeezing themselves. They can squeeze themselves and then they can move down through this pore. So that is the process of extravasation or intravasation. Both the jobs can happen. Now they can mix, and then finally these macrophages, they come here and they are mixed with the population. With this, we understood that magnetic nanoparticles can induce immune system to get into the cancer cell, and they can do the job of killing. So this is the way they attract immune cells. So here, please, if you can. So then what we did was we could find that there is these like tumor necrosis factor alpha, interleukin 12, CD86, iNOS, nitric oxide synthase, all those enzymes are getting activated. And this is a clear indication if these enzymes are getting activated, that means they're all M1 associated genes. And M1 macrophages are good macrophages. And later, second, we could find that all these M2 associated genes were suppressed because of the nanoparticles. So that was something which I learned, oh my God, there's something great. That these nanoparticles are doing a great job. They are not only helping in drug delivery, but instead they're activating the the immune cells so that the macrophages, the good macrophages grow, and then they start, and they also inhibit the bad macrophages. That's the job of magnetic nanoparticles. Fine. Next, please. Then the next job is, okay, this is talking about nanomedicine and immunotherapy. We created we try to understand what's happening in general. So what are these nanoparticles doing? They are doing a great job of, they will be nanoparticles with the drugs, all right? They will activate the macrophages. Macrophages will, and that to the good macrophages like M1. And then after that, they will be activating so many, the whole process. I mean to say they will be activating toll-like receptors, toll-like receptor 4, and then these toll-like receptors will do the job of T-cell priming, and then this T-cell priming will lead to, the clonal expansion of the T-cells will happen, and then those T-cells, we can get a help by killing. That means they will be doing the job of killing the tumor. So that's the idea. But this whole process can happen outside the body also. It's not necessary that we need to do the whole process inside. We can do this priming. This is known as priming. So we can do in outside the body. And by what? By using a chip. by using the cancer cells coming from the patient, by using the immune cells coming from the patient, and then we can mix all of them. We can induce this process of priming in the outside the body, and then get the clonal T cells from the body, from the chip, and then extract them and inject back into the body. So this is the way we can do the job of, priming outside the body. This is the idea, and that's the personalized approach. Next, please. So this is a process wherein we know that this is known as enhancing immune response, wherein there is an antigen. It's a common immunology. There is an antigen, and then that antigen will be induced, and then there will be like a dendritic cell or a macrophage will detect them, and then the dendritic cell will activate the T cells, and then T cell will then a kind of T helper cell, and the T helper cell will activate the cytotoxic T lymphocyte. So that is very common immunology, which we all must know. So then after that, there is also a process of reversing immune suppression. So there are, of course, I think it's too much of immunology. I understood that I don't have to teach a lot here. Next, please. So here we are talking about tumor on a chip platforms. Now, the actual topic comes. And what we are intending to do, if you all have understood, we have developed, now what have we understood till now? We have developed nanoparticles. We have attached drugs onto it. Then we have also attached targeted molecules like antibodies or folic acid. This whole thing will be transferred to a cancerous cell. And once the cancerous cell interacts with these nanoparticles with the drug, then they will get killed. Apoptosis will be generated. So this is happening normally. Okay, but it happens only in 2D system. But in animals, we could not get success. All right. And then we also studied how immunotherapy is useful and how is it working when we are using nanoparticles. Understood. Now, what happens when we are using, please go back. Here, what happens when we are using a 3D system, a 3D microfluidic system? In 3D microfluidic system, these are the in vitro models. What happens is, in 3D microfluidic system, is that the cells will not be having a good surface area. It will be in a condition wherein the cells will have a particular small space which will be exposed outside. And the whole area will not be exposed outside. So we have that very small space, okay, for the drugs to enter. So the probability of attacking these drugs with the nanoparticles onto the surface of the cancerous cell is less now. The probability has decreased. In 2D system, the probability was maximum. But in this kind of setup, the probability has decreased because it's a three-dimensional system, all right? What do you mean by three-dimensional? Now you might say that, come on, it's a three-dimensional system, that means the surface area increases. No. In this kind of 3D system, the cells are growing in 3 dimensions and having a very small area of exposure, all right? So that's the idea. That's the reason behind why we are telling this has to be 3D microfluidic system. Now here, what are the advantages of this system? In vitro, biomimetic tissue architecture and perfusion systems, all right? Then preservation of cell phenotype, heterotypic cell matrix, and of course, we can engineer the microenvironment, especially tumor microenvironment. We can engineer them. We can develop in such a fashion these micro-engineered system that we have to use all kinds of physics while developing the single channel, and then after that allow the cells and allow the nanoparticles to grow or to attack them. So that's the basic idea. Next, please. So how to fabricate this? It's a very simple structure. wherein we have a silicon wafer, and then we are making a mold. I think you all guys have made cakes in the house. Cakes, Hassan? Yes. So you have a mold, right? And in that mold you add something. Okay, first you must imagine you are creating a mold. Process. So we are actually creating a mold here. And then using that mold, this is like, this mold is, this is a master device. So we have created a mold like this. And then after that, we have this mold used for the whatever purpose you want. All right, next please. So this is also a 3D structure wherein we have all kinds of designs present like this. Next. So here, what we are doing is, this is a 3D system wherein the actual boring section or boring part of the lecture starts. So here, what happens is, there is a PDMS or a polydimethyl siloxane. All right. So this is a 3D system wherein we will be growing the cells in a very, very regulated fashion. All right. But before that, can we go ahead first or next? Yeah. So what happened is, in 3D microfluidic system, we have very, very regulated fashioned growth of the cancer cells and immune cells, very regulated fashion. But before this, what we have done is, we had made a spheroid. Spheroids or organoids. What is that? We're just taking the cancerous cells, which is mixed with tumor cells, everything will be there already. We take it from the patient and we grow it in a medium. Like we just allow them to grow, like a clone. You must have done cloning. Some clonal studies, some plant clones, you must have plantlets. PTC, you must be having plant tissue culturing. Yes. No? Yes. Maybe, okay, you must have heard about it. So there are clones which are formed. So in the same way, the cancerous clones are made and they are known as organoids. Or even sometimes like the smaller ones are known as spheroids and the bigger ones are known as organoids. So we were developing spheroids. Can you go ahead? Yes. So these are the different clones of the cancerous tissues. Now, what we did was, we took these tumors, they are patient sample. They're not just like that. They're all patient sample supplied by the nutrition hospital. So these are individual tumors, okay, but derived from the same patient's sample. And then what we did was, we wanted to study, previous part, yeah, this one. Yes. So what we wanted to study is, we wanted to tell the doctor, hey, we will tell you what particular drug you have to give and at what concentrations. So we told the doctor that we will tell you that. So here we have grown this spheroids and we use cisplatin, carboplatin, all these kinds of drugs. And then finally, these are all the stains, which confirms that these are the dead ones. These are the dead tissues. So with the doxorubicin 8.6 micromolar, carboplatin 538 micromolar. So these are the dead tissues. We just told the doctor, see, this is the drug and this is the report. Now you can give this thing to your patient. And then after that, he checked with the results coming from the patient, and he said that we are getting results. That's something great, because he was about to give him gemcitabine. He was about to give that particular drug, but we said, wait, before you give the drug, please give us some samples, and we can study that. All right, and within 8 days or 8 to 10 days it took, and then we could come to the conclusion, no, we can give this particular kind of drug. And this was all happening in Dr. Alejandro Centella's lab, which is in UNAM Biomedicine. So the whole work was done there before. Yes. So you can see the structures of the cells and we could confirm that the killing happens using this optical microscope. Then next. All right. So here in Siltech in Labman, what we are doing is we have been making this kind of 3-channel system. Now, what are these 3-channel systems? The thing is, in the body, there is a tissue, and that tissue is connected to a stromal system. Stromal system means blood capillary system. Now, the tissue which is connected to the blood capillary system, it will be interacting. I mean to say, this blood capillary system will have all sorts of blood cells, all sorts of neutrophils, macrophages, T cells, B cells, etc. All right. So then, what will happen is the tumor cell, the tissue microenvironment will be in tandem, which will be connecting in close contact with the stromal cells. Stromal cells is an endothelial cell coming from blood sample, like from blood. It's a connective tissue. And inside the blood, there will be all kinds of cells moving. So now that we would like to mimic. Here, what we have done is we have mimicked all 3 chambers. So one channel is of the tumor chamber. The middle one is actually, the middle chamber is actually the stromal cell, which is mimicking the endothelial cells of the blood capillary. And the last chamber, this one, is the immune chamber, where it will be growing the cancerous cells which are existing inside in the capillary. That's the idea. So we have this kind of model we have made, and then this was what are these 2? These are the molds, and finally we could make this kind of setup. It's looking really bad, but the thing is, we were interested in allowing, like creating a 3-chamber, but in connection with each other. There must be some or the other connection between each other. So you can see here, all these 3 are connected by very thin line compartmentation. Okay, there will be a compartmentalization. Okay, and they are like highly porous structures, so that there is continuous. For example, cytokines are made. Those cytokines will be passed on through those compartmentalization, or there will be something going on here and there. Next. So we have this kind of 3D microfluidic system. Then we wanted to study what kind of matrices, I mean to say, an extracellular matrix is required for the cancerous cells to grow. So we have used matrigel, collagen 1, BME2, RGR. These are the 3 different kinds of hydrogels on which the cancer cells are growing. Okay, it's actually mimicking the extracellular matrix in the body. All right, the next one. I think we are next. So what we have done here, you can see here the simple discrimination, or not the discrimination, a difference between a 2D and the 3D system. So you will find that in the 2D system, the viabilities, now here, you can see here. So there are the one paclitaxel olaparib, cisplatin, and the other is on different cell line, that is again cisplatin. So we did screening between the 2D system and the 3D system, and we could find, you can see here. So here we have, this is the 2D system, and this one is a 3D system. All the dark ones are the 3D systems. So you can find that the viability here, the viability in the 2D system is like, it's like 40% or 45%. And we could get here the viability approximately at 20 to 30%. So here you're getting almost similar. Here it's like going down, the viability goes down. So almost, we try to get the viability almost the similar kinds in all these kinds of cell lines. So that when we did screening of the drugs in such a way that in either 2D or 3D, in both the situations, which particular drug is killing the cells, all right? Which particular drug is the best one to kill the cell. And the result must match, like the 2D and 3D results must absolutely match. It should not be different. That's what our idea was. The next. And then you can see here, you can imagine that we can develop these many different kinds of chips in the body, or from the body, like eye on a chip, lung on a chip, vessel, liver, heart, triple B, intestine. All of these kind of chips can be developed, and for example, these all chips can be a representative of one individual in future. So in future, there are chances of having a bank. All right. And okay, in my bank, for example, Bank of Goldie Oza has got all these different kinds of chips. And before taking a pill, you will first inject whatever drug you would like to take. You will first inject into these chips and confirm and check with it whether it's not affecting any of these body organs. And if they are affecting the body organs, then you will skip those drugs and then you will go for another one. So like that, in future, we would like to have a kind of pathological system or a bank. All right. So this is the idea. And for example, I'm talking serious here. I am allergic to 5 drugs. Sure, I'm allergic right now. I'm allergic to penicillins and I'm allergic to many other drugs like sulfur. I'm allergic. So how could I know that? Previously, like even now, we take the drugs and then we feel like, oh my God, I'm getting some problems in the body, some flare-ups. And then immediately we'll rush to the hospital and then the doctor will ask you, which drug you have taken, my friend? I've taken this. Oh, you are allergic to this. So you are on the verge of death. On the verge of death, and you took those medicines, right? And then you finally understood. It happened with me when I was 10 years old, and my doctor was injecting me like penicillin, like as if it was a good playing game for him. He injected penicillin every time I used to go, and then suddenly I had a very terrible delayed hypersensitive reaction. I was admitted to the hospital for almost like 1 month, and then after that, the doctor is writing and telling me, my friend, you are allergic to these particular kinds of drugs. Never ever take in your life, otherwise you will be dead. So this was the statement given by him. But now, from now onwards or in future, we can have this kind of chip wherein we can first inject or insert this kind of drugs into these chips and check whether it's working or not. Everything is fine, and then we can use them. That's the idea. Next, please. Thanks a lot. And I think this is the way how the drugs, like we can, like a human body, we can have all the chips connected together, like a human body, and then inject the drug and then allow the drugs to move in all these organs. And then finally we can check, okay, what is the functionality of each and every organ based with the drugs which we have used. The next one. And then we have also done single cell analysis, wherein we are talking about how to identify, how to detect whether the cell is being killed or what kind of, like how can we detect a cancer cell in a blood sample. For example, now, there are patients, especially talking about, it's like my last part of the session, don't get annoyed. But then the thing is, we have leukemia patients. What happens in the initial stages is the leukemia cells are like approximately in numbers, right? Tens or hundreds, maximum hundreds or tens or 50s. These are the numbers, not high. So I just visited to one histopathology lab in Kirita, Japan. It's the only histopathology lab here, and they get samples from US. Why? Because they are specialists in identifying a leukemia cell, and that too from infants, and that too from children like 4 or 5 years old. So they're very specialized. And this guy told me that, my friend, we are facing one problem. We're using every time flow cytometries. And flow cytometries, they need approximately 10 raised to 6 cells. Okay, they have very less amount of, the sensitivity is less. We need at least 10 raised to 6 cells to identify, I mean to say, to get a proper number that, okay, this particular child has got particular cell, leukemia cell. If it is more than 100 or 1,000, then only the patient sample will be detected. I mean to say, it will come into the detection limit, and then you will get the answer. Okay, the person is suffering from leukemia or not. But what he said was, he needs a sample. Okay, he needs to detect at a very, very lower stage. If the patient is suffering from leukemia, immediately, I mean to say, even if there are 10 or 100 cells, the detection must happen. If it happens at that stage, the treatment strategies are there. But if it happens at a very, very advanced stage, the treatment strategies doesn't work. And that was the idea, the reason. And then we started working with Harvard and MIT mixed on this kind of nanowell system. It's a nanowell cytometry system. It's mimicking flow cytometry. But then in this kind of cytometry, what we could do is we just could identify 200 cells. Next, please. So this is a complete flow cytometric system, nanowell flow cytometry system, wherein we could identify 200 cells also. If there are 200 leukemia cells, we can identify. But since the topic is single cell analysis, we would like to go further, not 200, we would like to detect 20 cells. So I hope if we are successful, we can attack leukemia at a very, very early stage. With this, I would like to thank you all for listening to my work. I hope some of you have exited, but thanks to all of you guys who have exited as well as who are present here for having such a patience to listen to this. And I hope we all do a great job in future so that we can come up with some solutions to this dreadful cancer. That's what. Thank you, sir. Thank you. On one announcement, I have, if anybody is interested in organ ownership, we have in Cinetech, we have opened up a platform online lecture session. And from May, we are going to start organ ownership course sessions only for 5 days. All right, 2 hours each day. So in May, we are opening that. So maybe if anybody is interested, it's an online session. No, you don't have to travel all the way to Cinetech. So it's an online session. Anybody who is interested, you can like just check in Cinetech my Goldie Oza thing, and you can identify that. Right? Thanks a lot. Thanks.
Okay, so do you have any questions for Dr. Oza?
Yeah, please.
Yes, it was a really interesting talk. Thank you very much. I was wondering, you mentioned that the main problem with nanoparticles and 2D systems is the delivery system, right? That they don't reach. Because obviously in a 3D system, the physics and the microfluidics are completely different. Have you considered using to increase specificity virus-like particles, VLPs?
VLPs, all right.
Yes.
Actually, VLPs, we were planning to use with respect, there was one virus, I think rotavirus, and there is calicivirus. So this calicivirus, we wanted to, like, of course, we have used that. But I think it's being used in the presence of calcium, when the concentrations of calcium are very high, then these calicivirus will be releasing the drug effectively. And if the concentrations of calcium are not very high, then the release will not happen. All right, but yes, of course, that's a good plan. But yes, if you have something of that kind of setup, so what we can do in the future is we can have this organ on a chip model, and we can study it. That's my plan. That's the idea. that we use this kind of organ on a chip or a cancer on a chip model and use different kinds of drug delivery systems, like not only nano, but even the VLPs. Also, there are calixaries. There is I think Madam must be interested. Like there are medicinal drugs, of course, of rotaxanes or calixaries. These are the different kinds of structures which will be used in this kind of cancer on a chip. And then we will be like getting an idea, insights of how these, which one is best amongst all of them, and how it happens, what activation it does, whether it activates the immune. Because the idea is not just to kill the cancer cell. We have tried that a lot. Okay, killing is happening, fine. Not only that, we need to activate the immune system. And of course, VLPs are also very good in activating the immune system, and they do a great job because they are very efficient antigens. Yes, that's true. And that's the reason we would like to use them also in future in this kind of tumor on a chip models.
Thank you. Thank you.
Thank you.
So I have two questions. Is there any difference, or is more effective to use one method in one kind of tissue, and for example, for I don't know, for pulmonary cancer, if it's better to use one type of method than using, for example, another one for stomach cancer?
Yeah. The thing is, you're talking about two different types of cancers and same drug.
But then the metal and the
Nanoparticles? Yes. Okay, not only nanoparticles, it's actually drugs also. Like doxorubicin is an in general drug, universal drug. It has been used for, all kinds of, like almost like 7 to 8 types of cancers. And of course, these nanoparticles, they can be used, but then before that, before using any nanoparticle, we have to study the uptake. All right, uptake of the cancerous cells. Like for example, when you talk about gold nanoparticles, the uptake is efficient in very highly efficient in blood, pulmonary lung cancers, but the uptake in triple B is very less. So there we have to use a particular kind of polymer, which goes to like none of the metallic nanoparticles are useful whenever we need to transfer these nanoparticles in the triple B, like the blood-brain barrier. It's impossible. So we need to use a kind of polymers which can pass through it. So these, and that too through the nasal injection. So metallic nanoparticles are useless. But yes, we can use. We need to check the uptake first, and then after that use a particular kind of nanoparticle.
Do you consider the use of anti-cancer peptides as the treating agent?
Exactly. So these anti-cancer peptides, we have used microRNA, we have used the peptides, like even aptamers. Not only that, like aptamers are basically targeting agents. So we have used aptamers also. And whenever I was talking about the targeting agents, it's antibodies, it is aptamer, as well as it is like molecules, chemical molecules, biochemical. It's found.
I want to ask you, well, you were speaking about cancer in specific. Do you believe that it is possible to generalize or to have a kind of disease, the same technique? So instead of taking into consideration just cancer, other more general one, for sure, having chips for every single kind of
Exactly.
disease.
Actually, my problem is I've got the very bad habit of resolving only one problem at a time. But yet, but then the thing is, actually, the generic term which people are using is organ on a chip. I'm using here cancer on a chip. But the generalized term is on organ on a chip, wherein people have studied the problem of Alzheimer's, for example. So they have made a specific model for Alzheimer's. Though it's very difficult, but they have made partially, I should say. So for each disease, we can develop, and people are developing. For example, like University of Calgary, University of Caltech, and MIT, they are focusing on 5 different diseases. One, the neuronal diseases, they are making specific models for that. Then they are making especially models for eyes, some specific models. Then, of course, cancer on a chip is also included. Then the fourth one is talking, and that's the most difficult task which I was talking to you guys. That is the gut mucosal, mimicking mucosal system, and that to the gut intestinal tract on a chip. So intestinal tract on a chip, which is the most difficult organ on a chip to be developed till now. Like if it is developed, it will be the most difficult. It's really complex because in 2D system, if anybody must have worked, the biggest problem, if you grow a cancer, a normal cell or a cancer cell, the biggest problem is that they will be infected by bacteria always. And then what you will do, you will take those cells and put it in the dustbin because they are useless, because they are being infected by the bacteria. But what we are interested in doing this in intestinal tract on a chip is that we are interested in growing cells in tandem with the bacterial cells. And that's the biggest task ever, because it should not infect the intestinal cells, and it should stay also in that. So that's the most difficult task. But if we resolve that, all the problems of the microbiome with respect to that will be resolved. We know that to resolve this issue of any disease, we can have this kind of microbiome. We need to develop this kind of system. Even for diabetes, people say that the microbiome is responsible for that. So we can have those models also.
I have another question. Your opinion. How far are we from the substitution of clinical trials with these models?
Yes. Now let's talk about US. They are highly developed in that case. So they have there are two institutes at this moment. One is headed by Ali Kadem Hussein, and this guy he was from MIT, and then after that, now he has opened up this institution, his own institute, wherein he is taking samples from the patients and developing the chips, and then after that, doing the screening of the drugs, and then connecting with the hospitals. So this thing is going on, which has already started in US. And I think it will be in the next 3 to 4 years, this will be coming here also. So all over the world, this will be a common.
But also with new drugs, or just with already known?
All right, that's a good question. Thanks a lot, Matt. I forgot to address this. The thing is, all the companies, what they do, that they have to, for example, if they make a drug, they have to cross through all the clinical trials, like clinical trial 1, 2, 3, 4, and then finally, when it comes to the human patient, at that particular point, the drugs will not. Maybe the drug might fail at clinical trial 2 or clinical trial 3 itself, or it might enter into the human market, and then after that it fails. Most of the vaccines, we have seen that. So what is the advantage of this guy, this kind of organ on a chip is, we can make the organ on a chip models derived from humans, and then use the drug, which is at the clinical trial stage 1, or even if it has not even if it has not come to that stage, before that only we can use it in 3D systems, and we can see, okay, whether it is toxic or not. If it's not toxic, they can move ahead. This saves approximately billions of dollars of the industries.
I guess also 2 3 years.
Exactly. So because this is talking about whether it is toxic to immune cells, it's toxic to, and that to human immune cells. It's toxic to cancer, or it's toxic to any of the organs. So every system will be studied in detail. So this is a big market for the companies, the pharmaceutical companies. So thank you, ma'am. Thanks for your question. Thanks a lot.
I want to ask, how effective were those medicines in the nanoparticles when attacking the cancer stem cells? Because even if they're cancer stem cells, as far as I've read, they have like a certain tolerance to cytotoxic elements. So how effective are they in killing or reducing those cancer stem cells?
I'm sorry, I have not started any work on cancer stem cells because the problem with them is because growth of the cancer stem cell is a very difficult task. There was, of course, I was working, all this thing was done in Simstar when I was doing my postdoc, these things. So those people it was very difficult for them to grow the cells, cancer stem cells, and to maintain them. So we were like, I wanted to use these particles over there, but it was not possible. So that's the reason I think we need to work hard on that. And if anybody is successful, I would like to use my system in that kind of cancer stem cells. I'm sorry, I'm not done that.
I have a question regarding the drugs that we use for cancer treatment. So this is a very nice idea, very nice project, but what about the side effects that patients still have because of these drugs? Maybe they will be effective, but the side effects will be present. So have you thought about the possibility of exploring a new drug that doesn't have these side effects?
Actually, the thing is, the main idea behind this kind of drug delivery system was to decrease the side effect. All the nanoparticles have come up, even the VNPs, they are all responsible for making the the drugs more targeted and not affecting the normal cells. Because generally, people take doxorubicin directly and then they kill the normal cells as well as they kill the cancerous ones. So to avoid that, these nanoparticles we use. So now, further to avoid that, we have started using anti-aptamers or antibodies or any kind of biomolecules on this. Why? Because, for example, folic acids. The cancerous cell will express millions, we checked, it's like 10⁹ folic acid receptors. But in the case of a normal cell, folic acid receptors, though they are expressed in 10⁵ or 10⁵, but they will be always intracytoplasmic. They will not be exposed outside. They're always intracytoplasmic. So in the case of, that's the reason we started using folic acid so that they are becoming more targeted, because in cancer cells, it is always extracytoplasmic. That was the motive. So this was, hence this made our life easier that, okay, we'll use maybe glutathione and a kind of acidic pH, a kind of hypoxic environment. All these things are making it more targeted towards the tissue, which decreases the side effects of the drug. But yes, even now, as I told you, when we were dealing with the mice, with that little concentration of doxorubicin and with that little amount of the nanoparticles, also it was affecting the health of the mice. All right. So we could not at this stage, we could not become 100% targeted. So for that purpose, yes, we were using biomolecules, okay, or we were using some antibodies. But now aptamers have got a very high capacity of interacting with the cancerous cells. So now we are using aptamers to make it more finer, like fine targeted or synaptic targeted happens with that. So that's we are working on it.
Thank you. Any more questions?
Do you believe this technology will one day be part of general or large-scale clinical trials? And if so, what would you say are the major obstacles to reaching this goal?
Yes. First thing, of course, this will become the future, and this will be a part of all the hospitals, at least at this stage. I'm thinking, like we have a pathological lab. In future, this kind of thing will be existing in all the hospitals, because whosoever the doctors, they want to suggest in medicine, prescribe any medicine, they will tell the patient, okay, go to the lab and have your some part of your have your biopsy, have your certain cells given to them, and then they can have complete study. They will do the phenotypic study, they will do the genotypic study, and then they will also do all sorts of drug screening also on this kind of tissue. So this will become a future, of course. Then what are the problems? The biggest problem is that we are biopsy itself. Because we don't like to do this kind of, anybody is injecting some needle and then pricking some cells out of us. It's dangerous sometimes and it doesn't help us. So this will be the biggest drawback in future for this particular technology. But yes, in certain cases like cancer, for now, we are doing biopsy. It's like 100% required. So in cases which is very important, this has to happen. So we cannot avoid this. So this will be the barrier also, but this will be the solution also. So one and see. Yeah. And I think this will be the near future, next 3 to 4 years for sure. So like people will be knowing about it and everybody has to go for it. This will be the idea.
Okay. Let's see. Do you have any more questions?
So even if you have any questions, we can have an interaction from any time, whenever you want, we can. And I can share with you guys, maybe my email IDs, I think somehow, and we'll get it.
And also,
We have your email, so if you allow us to share it with. Please, sir, please, you're Gerardo. You're Gerardo? Yes, I am Gerardo. Finally, I will meet him also. Thank you. Thank you, so we can share. Thank you.
also you are free to give the invitation to join you as internship students or social service. If you
Yes.
want.
The thing is now this particular field needs human resources a lot. Now the thing is, as you have seen, there's so much of work required. So many organs on a chip is needed to be done. So I would request you guys that if anybody, now the guys are left very few, but if you can transfer this message to others also, that please, if anybody is interested in working with us in LabMin, you are please feel free to come over there and to visit our lab and to work on this particular topic. Though we are small at this stage, but then we need to grow at every point. So I would like to request you that if anybody wants to work for 3 to 6 months for any project, please feel free to come there and work with us. Okay.
Mr. Edgar is here with us. He's from the social service department.
Okay.
So at the end, we will have a bit of a conversation.
Thank you, sir. Thank you, ma'am.
Okay, so let's give another round of applause. And the line presentation. And on behalf of Arkansas State University, I would like to express our sincere gratitude for sharing your knowledge and insights about this topic. Thank you so much. We wish you all the best for your future projects.
Picture.
Oh, yeah. Yes.